before mammals ruled the land, before birds filled the skies, before humans ever walked upright, there were snakes, creatures born from an ancient experiment in evolution, a radical redesign of the vertebrate body. Limbs vanished, bodies elongated, muscle, bone, and instinct reshaped into something entirely new. For over 100 million years, snakes have mastered environments that challenge nearly every other predator on Earth. Deserts that scorch beneath relentless suns, rainforests layered in shadows and secrets, oceans where breath must be rationed with precision, mountains where cold tests survival itself. They move without legs, yet climb trees, swim oceans, burrow through stone, and strike faster than the human eye can perceive. They sense the world not only through sight, but through chemistry, vibration, and even heat itself. To some species, warm blood glows in darkness like a beacon. Their venom is not chaos. It is molecular engineering refined over millions of years. Proteins crafted by natural selection, precise enough to silence nerves, unravel blood, or dissolve tissue, and powerful enough to inspire both fear and medicine. They can swallow prey larger than their own heads, shut down their metabolism for months, survive in deserts, jungles, reefs, and frozen tundra. Some even reproduce without a mate. Across cultures, snakes have symbolized life and death, healing and destruction, wisdom and deception. But beyond myth, beyond fear, lies biology. In this video, we are not just listing facts. We are uncovering the science, the mechanics, the evolution, and the hidden systems that make snakes one of the most extraordinary lineages in vertebrate history. This is not just about reptiles. This is about survival, adaptation, power refined through time. These are 100 facts about snakes, and by the end, you will never look at them the same way again. In the primordial depths of Earth's evolutionary chronicle, approximately 100 million years ago during the Middle Cretaceous period, a remarkable transformation began to unfold within a lineage of lizards that would eventually redefine the very concept of terrestrial locomotion. These ancient reptiles, driven by selective pressures we can only partially reconstruct from fragmentary fossil evidence, embarked upon one of nature's most radical experiments in body plan modification. The ancestors of modern snakes gradually relinquished their limbs, not through catastrophic loss but through subtle, incremental changes spanning millions of generations, each modification conferring advantages in navigating increasingly specialized ecological niches. The fossil record, though tantalizingly incomplete, preserves crucial snapshots of this transformation, including specimens with vestigial hind limbs and pelvic girdles that hint at their four-legged ancestry. This evolutionary trajectory represents one of the most profound examples of morphological innovation in vertebrate history, demonstrating how natural selection can radically reshape body architecture when environmental pressures align with genetic possibilities, ultimately producing creatures capable of traversing environments that would remain inaccessible to their limbed relatives. The biomechanical implications of limblessness extend far beyond simple locomotion, creating a cascade of anatomical innovations that permeate every system of the serpentine body. Without appendages to manipulate their environment or defend territories, snakes developed elongated vertebral columns comprising between 200 and 400 vertebrae, each articulating with its neighbors through sophisticated ball and socket joints that permit extraordinary flexibility while maintaining structural integrity. This elongation necessitated profound reorganization of internal organs, which became dramatically attenuated and arranged in linear fashion rather than the compact clustering seen in most vertebrates. The right lung, for instance, extends through much of the body cavity in many species, while the left lung has either diminished to vestigial proportions or disappeared entirely, an asymmetry reflecting the spatial constraints imposed by an elongate body form. The heart, positioned strategically in the anterior third of the body, must generate sufficient pressure to perfuse tissues across remarkable distances, particularly in arboreal species where gravity opposes blood flow to the brain during vertical climbing. These cardiovascular adaptations include specialized valves and muscular modifications that would fascinate any comparative physiologist examining the limits of circulatory efficiency. The muscular architecture underlying serpentine locomotion represents a masterpiece of evolutionary engineering, comprising hundreds of discrete muscle segments arranged in overlapping layers that function as integrated antagonistic pairs. The epaxial muscles running along the dorsal surface work in concert with hypaxial muscles along the ventral and lateral aspects, generating the sinusoidal waves that propel snakes forward through what biomechanists term lateral undulation. Each muscle segment contracts sequentially, creating traveling waves that push against environmental irregularities, whether substrate particles, vegetation stems, or aquatic resistance. The efficiency of this system becomes apparent when one considers that snakes can achieve remarkable speeds despite lacking the lever arms that limbs provide to other animals. Studies employing high-speed videography and force plate analysis have revealed that the ground reaction forces generated during serpentine locomotion are distributed across multiple contact points simultaneously, effectively distributing propulsive effort across the body length rather than concentrating it in discrete limbs. This distributed force application explains why snakes can navigate surfaces that would challenge limbed animals, from loose sand to smooth vertical planes, adapting their locomotor strategy to match substrate properties with remarkable flexibility. Beyond lateral undulation, snakes have evolved several additional locomotor modes that expand their ecological versatility into environments where sinusoidal progression proves inefficient or impossible. Rectilinear locomotion, employed primarily by heavy-bodied species like pythons and boas when stalking prey or moving through confined spaces, involves sequential contraction of ventral muscles that lift and advance sections of the body in a caterpillar-like fashion, utilizing the broad ventral scales as anchoring points against the substrate. This mode permits nearly silent advancement, crucial for ambush predators approaching wary prey, and generates minimal visual disturbance compared to the conspicuous lateral undulations that might alert potential meals to danger. Sidewinding, perhaps the most visually striking locomotor adaptation, evolved independently in desert-dwelling species across multiple continents, demonstrating convergent evolution's power when confronted with similar environmental challenges. During sidewinding, the snake lifts loops of its body clear of the scorching sand while maintaining only 2 or 3 contact points, effectively minimizing heat transfer from the substrate while maximizing traction in loose, shifting sand that would frustrate other locomotor strategies. The resulting J-shaped tracks left in the sand appear almost calligraphic, recording the precise biomechanical choreography required to traverse one of Earth's most challenging terrestrial environments. Concertina locomotion, yet another variation in the serpentine movement repertoire, allows snakes to navigate narrow tunnels, crevices, and burrows where lateral undulation becomes geometrically impossible. In this mode, the snake anchors its posterior body against tunnel walls while extending its anterior portion forward, then anchors the front and draws the rear section forward in an accordion-like motion. This technique requires extraordinary muscular coordination and spatial awareness, as the snake must constantly assess tunnel dimensions and adjust its anchoring points to maintain purchase against confining surfaces. Researchers studying concertina locomotion in controlled laboratory settings have documented the sophisticated sensory integration required, as snakes must process tactile information from multiple body regions simultaneously while coordinating muscular contractions across segments that may be separated by considerable distances along the body axis. The metabolic cost of concertina locomotion exceeds that of other modes significantly, reflecting the mechanical inefficiency of repeatedly accelerating and decelerating body segments. Yet this energetic expenditure proves worthwhile when accessing subterranean refugia or pursuing fossorial prey that shelter in confined spaces inaccessible to surface-dwelling predators. The sensory apparatus of snakes has undergone equally remarkable modifications to compensate for limitations imposed by their unique body plan and to exploit opportunities available in their ecological niches. Vision in snakes varies tremendously across taxa, reflecting diverse evolutionary trajectories shaped by activity patterns and habitat preferences. Diurnal species often possess round pupils and excellent visual acuity, with some arboreal vine snakes developing horizontal keyhole-shaped pupils that enhance binocular overlap and depth perception, crucial for judging distances when striking at prey moving through three-dimensional canopy environments. Conversely, many nocturnal species exhibit vertical slit pupils that can close to pinpoint apertures in bright light, protecting sensitive retinal tissues while permitting rapid dilation for enhanced light gathering during nighttime activity. The absence of eyelids in snakes, replaced by transparent spectacle scales that fuse across the eye during embryonic development, creates the characteristic unblinking stare that has contributed to serpentine mystique throughout human cultural history, suggesting to observers an unwavering focus or preternatural awareness that has inspired both reverence and dread across millennia of human-snake interactions. Chemoreception in snakes represents perhaps their most sophisticated sensory modality, operating through dual systems that work synergistically to construct detailed chemical maps of the environment. The forked tongue, an icon of serpentine anatomy that has inspired countless metaphors for duplicity in human language, functions as a sophisticated chemical sampling device that collects airborne and substrate-bound molecules during its characteristic rapid flicking motions. Each tongue tip samples slightly different spatial locations, and the differential concentration of chemicals across the fork's tips provides directional information about odor sources, functioning analogously to binaural hearing in mammals where timing and intensity differences between ears permit sound localization. Upon retraction into the oral cavity, the tongue tips insert into the paired openings of Jacobson's organ, or the vomeronasal organ, located in the roof of the mouth. This chemosensory structure contains specialized receptor neurons that bind specific molecular configurations, transducing chemical information into neural signals transmitted to the accessory olfactory bulb of the brain. Through this system, snakes detect prey trails hours after the animal has passed, discriminate between potential mates based on pheromone profiles, and navigate complex terrain using chemical landmarks imperceptible to animals lacking comparable chemosensory sophistication. The thermal sensitivity exhibited by pit vipers and some pythons and boas represents a sensory innovation so extraordinary that it effectively grants these snakes a sixth sense unavailable to most other terrestrial vertebrates. The facial pits, positioned between eye and nostril in vipers or along the lip scales in pythons and boas, contain exquisitely sensitive thermoreceptive membranes suspended in air-filled cavities that function as biological infrared detectors. These organs can detect temperature differences as subtle as 3/1000ths of a degree Celsius, effectively imaging the thermal signatures of warm-blooded prey against cooler backgrounds. The neural architecture processing this thermal information parallels visual processing pathways, with thermoreceptive data projected to tactile regions of the brain where it integrates with visual input to create multimodal sensory representations of the environment. Behavioral studies have demonstrated that pit vipers can accurately strike at warm objects in complete darkness, guided solely by thermal cues, and can even detect thermal afterimages where warm-bodied prey recently rested, effectively reading thermal echoes of biological activity. This capability transforms nocturnal hunting from a challenging endeavor into a strategic advantage, as endothermic prey that might evade visual or chemical detection remain conspicuous through their thermal emissions, appearing to infrared-sensitive predators as glowing beacons against the ambient thermal landscape. The evolution of venom in snakes represents one of nature's most sophisticated biochemical innovations, transforming modified saliva into a complex molecular arsenal capable of subduing prey, defending against threats, and initiating digestive processes before ingestion occurs. Venom composition varies dramatically across snake lineages, reflecting different evolutionary trajectories and ecological specializations, yet all snake venoms share fundamental characteristics as complex mixtures of proteins and peptides rather than simple toxins. The venom of a single species may contain between 50 and 200 distinct protein components, each potentially affecting different physiological systems in envenomated organisms. This molecular complexity creates synergistic effects where individual venom components amplify or modulate the activities of others, producing pharmacological impacts that exceed the sum of individual effects. The evolution of venom delivery systems, from primitive rear-fanged mechanisms in many colubrid species to the sophisticated front-fanged apparatuses of vipers and elapids, demonstrates progressive refinement of envenomation efficiency. Front fangs in vipers can rotate forward from resting positions parallel to the jaw line, effectively deploying like biological hypodermic needles at the moment of strike, while the hollow or deeply grooved structure of these fangs permits rapid venom injection under muscular pressure generated by glands surrounding venom reservoirs. Neurotoxic venoms, characteristic of elipid snakes including cobras, krites, mambas, and coral snakes, target nervous system function through various mechanisms, though all ultimately disrupt neural signaling required for muscle contraction, respiration, and consciousness. Postsynaptic neurotoxins, among the most thoroughly studied venom components, mimic the structure of acetylcholine and bind irreversibly to nicotinic receptors at neuromuscular junctions, blocking legitimate neurotransmitter binding and preventing muscle fiber activation. As these toxins accumulate at motor endplates throughout the body, progressive paralysis ensues, typically beginning with cranial muscles controlling eye movement and swallowing before advancing to respiratory muscles. Presynaptic neurotoxins employ a different strategy, preventing acetylcholine release from nerve terminals by disrupting vesicle fusion or calcium channel function, effectively silencing neurotransmission at its source. The paralytic effects of neurotoxic envenomation progress with terrifying inexorability, as conscious victims experience progressive loss of motor control while remaining fully aware, a scenario that has inspired profound dread throughout human history and contributed to the mythological association of serpents with death and the underworld. Treatment requires mechanical ventilation to maintain respiration while antivenom antibodies neutralize circulating toxins and bound toxins eventually dissociate from receptors, a process that may require days or weeks depending on envenomation severity. Hemotoxic venoms, prevalent in viper species including rattlesnakes, copperheads, and Old World vipers, orchestrate systemic disruption of blood homeostasis through multiple concurrent mechanisms affecting coagulation cascades, vascular integrity, and cellular components. Procoagulant toxins activate clotting factors, inducing rapid consumption of fibrinogen and other clotting proteins in a phenomenon termed consumption coagulopathy, paradoxically rendering victims unable to clot effectively once clotting factors become depleted. Hemorrhagic toxins, particularly the snake venom metalloproteinases, degrade basement membrane proteins surrounding blood vessels, compromising vascular integrity and allowing blood to leak into surrounding tissues, manifesting as spectacular ecchymosis and tissue edema at envenomation sites. These metalloproteinases represent evolutionary modifications of endogenous matrix metalloproteinases that normally function in tissue remodeling, demonstrating how natural selection co-opts existing genetic machinery for novel purposes when selective pressures favor new functional capabilities. The resulting envenomation syndrome combines local tissue destruction with systemic coagulopathy, cardiovascular shock, and potential renal failure as degraded tissue products overwhelm kidney filtering capacity, creating a medical emergency requiring rapid administration of specific antivenom to neutralize toxins before irreversible organ damage occurs. Cytotoxic venom components, particularly phospholipids that catalyze membrane lipid hydrolysis, induce direct cellular damage at envenomation sites, destroying muscle fibers, connective tissue, and vascular elements in expanding zones of necrosis that may require extensive surgical debridement to prevent systemic infection and facilitate healing. The progression of cytotoxic effects follows a characteristic temporal pattern, with immediate pain and swelling giving way over hours to blistering and eventually frank tissue death as cellular destruction outruns vascular perfusion capacity. Secondary bacterial infection of necrotic tissue remains a significant complication, as devitalized tissue provides ideal culture medium for opportunistic pathogens that may seed bloodstream infections if not promptly addressed through surgical intervention and antimicrobial therapy. The ecological function of cytotoxic components remains debated, as tissue destruction seems excessive for prey subjugation alone, suggesting possible roles in predator deterrence or extraoral digestion that begins breaking down prey tissues before swallowing occurs. Some researchers propose that venom complexity reflects ongoing evolutionary arms races between snakes and their prey, with prey populations evolving resistance to specific toxin classes and snakes responding by elaborating additional venom components, creating an escalating cycle of adaptation and counter-adaptation played out across geological timescales. The feeding biology of snakes encompasses some of the most remarkable adaptations in vertebrate natural history, permitting these limbless predators to consume prey items that may exceed their own head width by several-fold through mechanical innovations that would seem physically impossible to observers unfamiliar with serpentine anatomy. The snake's skull represents a masterpiece of evolutionary engineering for kinesis, comprising numerous separate bones connected by elastic ligaments rather than the rigid sutures characterizing most vertebrate skulls. This construction permits extreme flexibility, with upper and lower jaws capable of independent movement and the two halves of the lower jaw connected anteriorly by an elastic ligament rather than a rigid symphysis. During feeding, snakes employ alternating movements of left and right mandibular and maxillary bones, effectively walking their jaws over prey in a ratcheting motion that gradually engulfs items far larger than the skull's resting dimensions would suggest possible. The quadrate bones, connecting lower jaws to the cranium, can rotate extensively, effectively extending jaw gape by projecting the mandibles laterally and ventrally to accommodate bulky prey. This kinetic skull architecture, combined with elastic skin between mandibles and highly distensible esophageal walls, permits some python species to swallow prey exceeding their own body mass, including ungulates and large primates that represent extraordinary energetic windfalls worth the considerable mechanical challenges their consumption presents. The digestive physiology of snakes has evolved to match their feast or famine lifestyle, with prolonged fasting periods punctuated by massive meals requiring extraordinary digestive capacity to process. During extended fasting, metabolic rate declines to minimal levels as energetic maintenance costs are reduced through downregulation of numerous physiological systems, including profound atrophy of intestinal mucosa as cellular populations turn over without replacement. This energy-conserving strategy permits snakes to survive months or even years between meals in extreme cases, though most species feed more regularly when prey remains available. Following ingestion of large prey, metabolic rate increases dramatically, sometimes exceeding resting rates by 20-fold in what physiologists term specific dynamic action, reflecting the energetic costs of regenerating digestive tissues, secreting digestive enzymes, and processing absorbed nutrients. The intestinal epithelium undergoes rapid hyperplasia, doubling or tripling in mass within 48 hours as cellular proliferation regenerates absorptive capacity. Digestive enzymes, including proteases, lipases, and nucleases, are secreted in massive quantities, creating a biochemical environment within the stomach and intestines capable of degrading even recalcitrant tissues like bone, feathers, and keratinous structures that would resist digestion in most other vertebrates. The capacity of snake digestive systems to process bone represents a particularly impressive biochemical feat, as skeletal elements contain minerals and structural proteins that pose significant digestive challenges. Gastric pH in feeding snakes declines to extraordinarily acidic levels approaching pH 1, creating conditions that solubilize calcium phosphate minerals comprising bone matrix while denaturing collagenous proteins that form bone's organic scaffold. The secretion of such concentrated hydrochloric acid requires substantial energetic investment and specialized epithelial cells capable of withstanding the corrosive environment they create, yet this capability permits complete prey utilization, extracting nutrients from skeletal tissues that would otherwise pass through the digestive tract unprocessed. The only materials consistently resisting digestion are heavily keratinized structures like claws, hooves, and certain feathers, which are eventually regurgitated as compact pellets after nutrient absorption concludes. This near-complete prey processing minimizes waste production and maximizes energetic return on the substantial investment required to capture, subdue, and process large prey items, an efficiency crucial for animals whose feeding opportunities may be separated by considerable temporal intervals. Temperature profoundly influences all aspects of snake physiology, as these ectothermic organisms lack the metabolic capacity to generate sufficient heat to maintain stable body temperatures independent of environmental conditions. Behavioral thermoregulation becomes essential, with snakes actively seeking thermal microenvironments that elevate or reduce body temperature as physiological demands dictate. Basking behavior, where snakes position themselves in sunlight to absorb radiant energy, represents the primary mechanism for thermal elevation, though the effectiveness of basking depends on numerous factors including substrate conductivity, air temperature, wind speed, and body size. Large-bodied species with low surface-to-volume ratios warm slowly but maintain elevated temperatures longer than small species that heat rapidly but also lose heat quickly, creating size-dependent differences in optimal thermoregulatory strategies. The thermal sensitivity of biochemical reaction rates means that physiological processes including digestion, locomotion, sensory acuity, and immune function all operate optimally within preferred temperature ranges, typically between 28 and 32 degrees Celsius for most temperate and tropical species, though preferences vary considerably across taxa adapted to different thermal regimes. Thermoregulatory behavior intersects with virtually every other aspect of snake ecology, constraining activity patterns, habitat selection, and even social interactions around thermal considerations. In temperate regions, snakes must locate suitable hibernacula for winter dormancy, sites offering protection from freezing temperatures that would prove lethal to organisms lacking antifreeze proteins or other cold tolerance mechanisms. These hibernation sites often support communal aggregations of hundreds or even thousands of individuals, particularly in northern latitudes where suitable refugia are spatially concentrated and thermally adequate sites are scarce. The den emergence in spring triggers spectacular mating aggregations in some species, most famously the red-sided garter snakes of Manitoba, Canada, where thousands of males converge on emerging females in writhing mating balls that have become iconic in popular depictions of serpentine reproduction. The selective pressures favoring communal denning likely include thermal benefits of aggregation, as grouped snakes reduce individual heat loss through mutual insulation, though information transfer about den locations across generations also plays crucial roles, with philopatric individuals returning to natal dens throughout their lives. Reproductive strategies in snakes encompass remarkable diversity, ranging from oviparity where eggs are deposited and develop externally through ovoviviparity where eggs are retained and hatch internally to true viviparity with placental-like exchange between maternal and embryonic tissues. Oviparous species must locate appropriate nest sites offering thermal and hydric conditions conducive to embryonic development, as eggs cannot regulate their own temperatures and remain vulnerable to desiccation if moisture balance becomes disrupted. Some python species exhibit maternal brooding, coiling around their eggs and generating metabolic heat through rhythmic muscle contractions that elevate clutch temperature by several degrees Celsius above ambient conditions, representing a rare example of endothermic heat production in typically ectothermic organisms. This parental investment extends over the entire incubation period, sometimes exceeding 2 months, during which the brooding female remains with her eggs, foregoing feeding opportunities and exposing herself to potential predation while ensuring optimal developmental conditions for her offspring. The energetic costs of brooding are substantial, with females losing significant body condition during the brooding period, yet the survival benefits to developing embryos apparently outweigh these maternal costs, as the behavior persists across multiple python lineages. Viviparous species, which include numerous viper and sea snake species, eliminate nest site requirements by retaining developing young internally until parturition, providing thermal stability and protection from predators throughout development. The transition to viviparity has evolved independently multiple times within snake phylogeny, suggesting strong selective advantages under certain ecological circumstances, particularly in cool climates where suitable nest temperatures remain difficult to achieve externally or in aquatic environments where terrestrial nesting would require females to abandon their marine habitats. The placental structures developed in some viviparous species facilitate gas exchange, nutrient transfer, and waste removal between maternal and embryonic circulatory systems, though these placentae are structurally simpler than mammalian counterparts and provide less extensive physiological support. Neonates born through viviparity emerge fully developed and immediately capable of independent existence, though their survival remains precarious as numerous predators, including adult conspecifics in some species, readily consume vulnerable juveniles despite their often potent defensive capabilities, including functional venom systems in many taxa. The diversity of defensive strategies employed by snakes reflects the intense predation pressure these reptiles face from numerous vertebrate predators including raptors, mammalian carnivores, and other snakes. Cretaceous represents the first line of defense for most species, with cryptic coloration and pattern-matching substrate characteristics rendering motionless snakes nearly invisible to visual predators. Disruptive coloration patterns featuring contrasting bands or blotches break up body outline, frustrating predator recognition even when substrate matching remains imperfect. When Cretaceous fails and predators approach dangerously close, many species employ dramatic behavioral displays designed to startle or intimidate potential threats. Rattlesnakes vibrate specialized tail segments composed of loosely interlocking keratin rings, generating characteristic buzzing warnings that advertise their defensive capabilities to approaching threats, an example of aposematic signaling where dangerous prey announce their status rather than remaining cryptic. Cobras rear up and expand cervical ribs to create intimidating hoods, while some species can project venom from forward-facing fang openings, accurately spitting at potential threats from distances exceeding 2 meters, aiming specifically at eyes where venom causes immediate pain and potential permanent damage. Hognose snakes employ elaborate death-feigning displays when threatened, writhing violently as if experiencing convulsions before rolling onto their backs with mouths agape and tongues lolling, presenting a convincing simulation of death that apparently deceives some predators into abandoning attacks on apparently defunct prey. The commitment to this display can be remarkable, with disturbed hognoses persistently returning to their ventral-up posture if righted by curious observers, maintaining the deception even when their persistence reveals the charade. The ecological effectiveness of Thanatosis presumably depends on predators preferentially selecting live prey over carrion, a reasonable assumption for many predators that may associate non-moving prey with disease risk or decomposition that renders carcasses less nutritionally valuable than fresh kills. Other species employ tail displays that divert predatory strikes away from vulnerable heads toward expendable tail tips, with some taxa possessing brightly colored tail tips that contrast dramatically with body coloration, effectively advertising them as targets. Tail autotomy, though less common in snakes than in many lizards, occurs in some species where tail loss permits escape from predator grasp, though the inability to regenerate lost tail segments makes this defense costlier for snakes than for lizards that can regrow severed tails. The relationship between body size and prey preference in snakes demonstrates strong scaling relationships, with ontogenetic dietary shifts common as growing individuals graduate from small ectothermic prey to larger endothermic targets that provide greater energetic return per capture but also pose increased handling challenges. Neonatal snakes often specialize on invertebrate prey including insects, spiders, and earthworms that adults would ignore as energetically unprofitable given their handling costs, though these small prey items match the gape limitations and energetic requirements of recently hatched individuals. As snakes grow, their feeding apparatus enlarges proportionately, permitting capture and consumption of progressively larger prey while simultaneously increasing energetic requirements that make small prey items insufficient to meet metabolic demands. Some species maintain dietary specialization throughout ontogeny, with certain snake-eating specialists consuming only other snakes throughout their lives, while others exhibit dramatic dietary shifts, transitioning from lizard specialists as juveniles to rodent predators as adults. These ontogenetic transitions can occur quite abruptly, sometimes spanning only a few months as rapidly growing individuals cross size thresholds permitting exploitation of new prey categories. Dietary specialization in snakes reaches extraordinary extremes in some lineages, with taxa evolving morphological and physiological adaptations permitting exclusive consumption of particular prey types that would challenge or poison generalist predators. Egg-eating snakes possess highly modified vertebrae featuring ventrally projecting hypopophyses that protrude into the esophagus, effectively creating internal eggshell crushers that pierce ingested eggs and permit extraction of liquid contents while compressed shells are subsequently regurgitated. The cranial anatomy of egg-eating specialists includes reduced or absent teeth, as these structures would impede egg ingestion and serve no function when prey requires no subjugation. Mollusk specialists feeding on snails and slugs have evolved asymmetrical jaws with more numerous teeth on the right mandible than the left, facilitating extraction of gastropod soft tissues from their shells through coordinated right-sided biting and left-sided bracing that effectively corkscrews prey from their protective calcium carbonate housing. These specialists possess modified saliva containing compounds that interfere with gastropod mucus adhesion, reducing the effectiveness of mucus-based defensive secretions that deter many potential mollusk predators. Aquatic and marine snakes have undergone profound modifications, adapting them for fully aquatic or semi-aquatic existences, with true sea snakes representing some of the most thoroughly marine-adapted reptiles, having lost the capacity to move effectively on land and giving birth to live young at sea rather than returning to terrestrial environments for reproduction. The body forms of sea snakes reflect their pelagic lifestyles, with laterally compressed paddle-like tails providing propulsive thrust and dorsally positioned nostrils permitting breathing while remaining nearly submerged. The venoms of sea snakes rank among the most potent known, featuring primarily neurotoxic components that rapidly immobilize fish prey, preventing escape into reef structures where dead or dying prey would remain inaccessible. The salt glands located beneath the tongue in sea snakes permit excretion of excess salt consumed incidentally during feeding and drinking, maintaining osmotic balance despite living in hypersaline environments that would dehydrate terrestrial reptiles lacking such specialized excretory capabilities. The single lung extends through nearly the entire body cavity in sea snakes, maximizing respiratory surface area and providing buoyancy control analogous to swim bladders in fish, permitting precise regulation of vertical position in the water column without continuous swimming effort. The evolution of arboreal specialists among snakes demonstrates convergent evolution of similar morphological features across distantly related lineages confronting comparable selective pressures in three-dimensional canopy environments. Arboreal species typically exhibit elongate, slender body forms with enhanced vertebral flexibility permitting navigation of discontinuous substrates where individuals must bridge gaps between branches that would constrain shorter, stockier species. Prehensile tails evolved independently in numerous arboreal lineages, providing additional anchoring points that effectively increase stability and reach while moving through complex arboreal environments. The ventral scales of many arboreal species feature enhanced keeling or angular modifications that increase friction against bark surfaces, improving purchase on vertical or inverted substrates where gravitational forces oppose adhesion. Coloration in arboreal specialists often tends toward green or brown tones matching foliage and bark, though some species exhibit elaborate patterns suggesting leaf venation or lichen incrustations that enhance crypsis when draped across branches or coiled in foliage masses. Fossorial adaptations in burrowing snakes reflect yet another set of convergent morphological solutions to life underground, where visual cues become largely irrelevant and tactile and chemical senses dominate sensory perception. Fossorial species typically possess reduced eyes covered by enlarged scales, reflecting relaxed selection for visual acuity in perpetually dark subterranean environments where elaborate eyes would provide little selective advantage while remaining vulnerable to abrasive damage during tunneling. The skulls of burrowing species are heavily ossified and compact, providing rigid digging instruments capable of forcing passages through compacted soil without sustaining structural damage. The scales covering the head in many fossorial species exhibit enhanced hardening and reduced overlap, effectively creating a unified bony shield rather than the flexible scale covering characterizing surface-dwelling species. Body cross-sections in burrowing specialists tend towards cylindrical rather than the dorso-ventrally compressed profiles of terrestrial species, permitting rotation within burrows while maintaining structural support against collapsing tunnel walls. The geographic distribution of snakes spans all continents except Antarctica, though their diversity peaks in tropical regions where warm temperatures permit year-round activity and high prey availability supports diverse assemblages. Latitudinal diversity gradients in snakes follow patterns common across numerous taxa, with species richness declining steeply with increasing latitude as seasonal temperature fluctuations intensify and growing seasons contract. The absence of snakes from certain oceanic islands, particularly those that never connected to continental landmasses, reflects dispersal limitations in organisms lacking the flight capabilities of birds or the marine colonization abilities of sea turtles. The presence of snakes on other remote islands, including the iconic flying snakes of Southeast Asian archipelagos, demonstrates that some species can disperse across substantial oceanic barriers, likely rafting on floating vegetation during storms or riding ocean currents to colonize distant shores. The evolutionary radiation of snakes on continents following the breakup of Gondwana has created distinct regional faunas characterized by different dominant families, with colubrid snakes dominating most regions, elapids prevalent in Australia, and vipers restricted to the Old World and Americas. The coevolutionary dynamics between snakes and their prey have generated remarkable examples of reciprocal adaptation, with prey populations evolving resistance to venom toxins and morphological defenses against predation, while snakes counter with enhanced venom potency and modified hunting strategies. Ground squirrels in rattlesnake endemic regions have evolved partial resistance to venom toxins, requiring substantially higher venom doses for immobilization than closely related species from snake-free regions, demonstrating geographically localized adaptation driven by sustained predation pressure. These resistant squirrel populations also exhibit behavioral modifications, including tail flagging displays that advertise vigilance to stalking rattlesnakes and harassment behaviors where squirrels approach and harass rattlesnakes, disrupting their ambush tactics. The heating of squirrel tails during these confrontations, detected by rattlesnake thermal sensors, apparently signals the squirrel's awareness of snake presence, as snakes rarely strike at tail-flagging individuals compared to squirrels unaware of lurking predators, suggesting sophisticated signaling exchanges between predator and prey that influence hunting success rates. Ophiophagy, or snake eating by other snakes, represents a fascinating trophic specialization occurring across multiple snake families, with king snakes and king cobras achieving particular renown for their ophiophagous habits. The immunological resistance to heterospecific snake venoms exhibited by some ophiophagous species permits them to consume venomous prey without succumbing to envenomation, though the molecular basis of this resistance varies across taxa. Some snake-eating specialists possess modified serum proteins that bind and neutralize venom toxins, while others exhibit receptor modifications that reduce toxin binding affinity, conferring resistance through reduced target sensitivity rather than toxin neutralization. The ecological implications of ophiophagy include top-down regulation of snake community structure, as dominant ophiophagous species may suppress populations of competing snakes, shaping community composition through intraguild predation. The evolution of ophiophagy may be facilitated by the relatively large meal sizes that adult snakes represent, providing substantial energetic returns that offset the risks and handling challenges associated with subduing elongate, potentially dangerous prey. Mimicry systems involving snakes provide compelling evidence for predator-mediated selection on coloration patterns, with harmless species evolving color patterns resembling those of sympatric venomous models, presumably gaining protection from predators that have learned to avoid the dangerous models. Coral snake mimicry represents the most extensively studied example, with numerous harmless colubrid species exhibiting red, yellow, and black banding patterns similar to sympatric coral snakes, though distinguishing features exist that permit discrimination by sufficiently observant individuals. The various mnemonics developed to separate venomous coral snakes from harmless mimics, such as red touch yellow, kill a fellow, red touch black, venom lack, reflect patterns in North American species but fail in other regions where different color sequence variations occur. The effectiveness of mimetic protection depends critically on predator learning, requiring that predators encounter models frequently enough to associate color patterns with unpleasant consequences, but not so frequently that mimics are common relative to models, diluting the protective benefit. Batesian mimicry, where palatable mimics resemble unpalatable or dangerous models, can become destabilized if mimic frequency becomes too high, as predators that encounter mimics more often than models learn that the warning pattern does not reliably predict danger. The cultural significance of snakes in human societies spans the full spectrum from sacred reverence to visceral horror, reflecting the complex and often contradictory relationships humans have maintained with these reptiles throughout history. Ancient Mediterranean and Middle Eastern cultures frequently associated serpents with healing and regeneration, possibly inspired by their periodic skin shedding that suggests renewal and transformation, with the rod of Asclepius featuring a snake coiled around a staff persisting as a symbol of medicine into modern times. Hindu mythology elevates the cobra to divine status, with Nagas representing powerful serpent deities associated with water, fertility, and protection, receiving worship at temples throughout South and Southeast Asia where live cobras may be maintained as sacred residents. Conversely, Abrahamic religious traditions often cast serpents as embodiments of evil or temptation, most famously in the Genesis narrative where the serpent's deception precipitates humanity's expulsion from paradise, establishing an antagonistic archetype that has influenced Western cultural attitudes towards snakes for millennia. The neurobiology underlying ophidiophobia, or fear of snakes, has attracted considerable research attention from evolutionary psychologists investigating whether humans possess innate predispositions towards snake detection and avoidance. Some evidence suggests that humans, particularly young children, more rapidly detect and attend to snake images in visual search tasks compared to other objects or animals, potentially reflecting evolutionary preparation shaped by the threats venomous snakes posed throughout human ancestry in African savannas where hominids evolved. However, the universality and innateness of snake fear remains debated, with cross-cultural studies revealing substantial variation in emotional responses to snakes correlated with cultural context and individual experience rather than purely innate templates. The disproportionate fear that snakes inspire relative to statistically more dangerous entities like automobiles or processed foods might reflect evolved threat detection systems that remain calibrated to ancestral hazards rather than novel dangers characteristic of modern environments, creating persistent mismatches between perceived and actual risk in contemporary contexts. Venom research has generated profound insights into neurobiology and pharmacology while producing therapeutically valuable compounds, demonstrating how organisms that inspire dread can simultaneously advance human welfare through scientific investigation. The isolation and characterization of alpha-bungarotoxin from banded kite venom provided the first high-affinity ligand for acetylcholine receptors, revolutionizing neuromuscular research by permitting receptor localization, purification, and molecular characterization that would have remained impossible without this fortuitously discovered molecular probe. Subsequent discoveries of additional venom-derived research tools and therapeutic leads have solidified snake venoms as extraordinarily rich sources of bioactive compounds, with current pharmaceutical development efforts targeting venom components for treating hypertension, thrombosis, pain, and cancer. The blood pressure medication captopril, one of the first angiotensin-converting enzyme inhibitors developed, originated from studies of bradykinin-potentiating peptides isolated from Jararaca pit viper venom, demonstrating how evolutionary experimentation over millions of years has generated molecular diversity unmatched by human chemical synthesis capabilities. The conservation status of many snake species has become increasingly precarious as habitat destruction, climate change, persecution, and collection for the pet trade compound to threaten populations worldwide. Habitat fragmentation poses particular challenges for snakes, as many species maintain large home ranges and undertake seasonal migrations between hibernacula, foraging areas, and reproductive sites, requiring landscape connectivity that becomes disrupted by human infrastructure development. Road mortality claims substantial numbers of snakes annually, with some species experiencing population-level impacts where road densities exceed threshold values, effectively severing metapopulation connectivity and isolating remnant populations in habitat patches too small to maintain viable numbers long-term. Climate change projections suggest distributional shifts for many temperate zone species as thermal regimes shift poleward, though whether species can track shifting climatic envelopes depends on dispersal abilities, habitat availability, and rates of evolutionary adaptation to novel conditions, creating considerable uncertainty about future snake biodiversity patterns. The deliberate persecution of snakes, motivated by fear and misunderstanding, continues to claim countless individuals annually despite growing recognition of their ecological importance as both predators and prey in food webs. Educational initiatives highlighting the role of snakes in controlling rodent populations and the statistical rarity of serious snakebite incidents aim to shift public perception from reflexive elimination toward tolerance and coexistence, though entrenched cultural attitudes and genuine concerns about human safety complicate conservation messaging. The development of bite-proof footwear and improved access to anti-venom have reduced snakebite mortality significantly in many regions, potentially reducing human-snake conflict by minimizing the consequences of chance encounters while field workers conduct agricultural or forestry activities. Protected area establishment remains crucial for snake conservation, providing refugia where populations can persist without direct persecution, though many protected areas are too small or isolated to maintain viable populations of wide-ranging species indefinitely. The biomimetic potential of snake-inspired engineering has begun attracting attention from roboticists and materials scientists seeking to replicate serpentine locomotor capabilities and sensory systems in artificial systems. Snake-like robots capable of navigating rubble piles during search and rescue operations, inspecting sealed pipes, or conducting medical procedures within body cavities demonstrate practical applications of bio-inspired design informed by understanding snake morphology and locomotion. The climbing abilities of snakes have inspired adhesive surface designs featuring directional friction asymmetries that grip effectively during forward motion while releasing easily during backward sliding, mimicking the function of snake ventral scales on vertical surfaces. Infrared detection systems inspired by pit viper thermal sensitivity have potential applications in night vision technology and autonomous vehicle navigation, though current artificial thermal sensors remain substantially inferior to biological counterparts in sensitivity and temporal resolution, highlighting the engineering sophistication achieved through evolutionary optimization. Climate-driven phenological shifts affect snake populations through multiple pathways, including altered timing of emergence from hibernation, mismatches between snake activity periods and prey availability, and modified interspecific interaction networks as species respond asynchronously to changing conditions. Earlier spring emergence driven by warming temperatures may expose snakes to late-season freezing events that historically occurred before emergence, increasing mortality risk during vulnerable post-hibernation periods when individuals are energetically depleted. Shifts in precipitation patterns affect snake distributions directly through altered availability of moisture refugia and indirectly through effects on prey populations, with drought-induced prey scarcity forcing snakes to expand foraging ranges or accept suboptimal prey that may provide insufficient nutrition for successful reproduction. The complex interactions between temperature, precipitation, vegetation dynamics, prey availability, and predation pressure create nonlinear responses to climate change that challenge predictive modeling efforts, though scenarios universally suggest significant redistributions of snake diversity across landscapes over coming decades. The role of snakes in ecosystem function extends beyond their direct trophic impacts as predators and prey, encompassing influences on nutrient cycling, seed dispersal, and disease dynamics that remain incompletely characterized. The deposition of snake excreta and carcasses returns nutrients sequestered in prey biomass to ecosystems, with nutrients becoming available to plants and decomposers, supporting productivity in nutrient-limited environments. Some snakes incidentally disperse plant seeds through consumption of frugivorous prey containing viable seeds that pass through snake digestive systems unharmed, though the ecological significance of such secondary dispersal remains poorly quantified. The regulation of rodent populations by snake predation may influence disease transmission dynamics in zoonotic pathogens maintained in rodent reservoirs, potentially affecting human disease risk in ways that become apparent only when snake populations decline and rodent abundances increase above historical norms, releasing disease dynamics from predator-mediated regulation. Neuromuscular adaptations permitting strike performance in snakes represent some of the fastest movements in vertebrate biology, with rattlesnakes accelerating their heads at rates exceeding 50 meters per second squared, comparable to accelerations experienced by fighter pilots during high-G maneuvers. These extraordinary accelerations require specialized muscle physiology and skeletal modifications that withstand tremendous forces without structural failure, as the accelerative phase of the strike lasts only 50 to 70 milliseconds, compressing enormous momentum changes into minuscule temporal windows. The sensory integration required for accurate strikes demands precise coordination of visual, thermal, and chemical inputs processed through neural circuits that compute prey location, velocity, and trajectory in real time, generating motor commands that aim the strike toward predicted interception points rather than current prey positions. The neural processing underlying this predictive targeting remains incompletely understood, though comparative studies suggest that specialized tactile and cerebellar circuits integrate multisensory information to generate appropriately timed and directed motor outputs, achieving hit rates exceeding 90% against moving targets in some species. The economic impacts of snakes encompass both costs associated with snakebite treatment and benefits derived from rodent control services, creating complex cost-benefit calculations that vary geographically and temporally depending on snake and prey densities, agricultural practices, and healthcare infrastructure. Agricultural regions supporting high snake densities may benefit from reduced crop losses to rodent pests that consume or contaminate stored grain, with economic analyses suggesting that ecosystem services provided by snake-mediated pest control can substantially offset snakebite treatment costs where both factors are quantified. The development of improved antivenoms with enhanced efficacy and reduced adverse reaction profiles could further shift cost-benefit ratios by reducing treatment complications and long-term morbidity associated with envenomation, improving quality of life for snakebite survivors while simultaneously reducing healthcare expenditures on managing complications. Public health interventions addressing snakebite risk remain underfunded relative to disease burden in many tropical regions where agricultural workers face daily exposure to venomous species, creating preventable mortality and morbidity that burden already strained healthcare systems. The molecular evolution of venom genes reveals patterns of rapid diversification through gene duplication, positive selection, and regulatory modification that have generated extraordinary toxin diversity from ancestral proteins with conserved physiological functions. Venom phospholipids, for example, evolved from housekeeping enzymes involved in membrane lipid metabolism through gene duplication events that released duplicated copies from purifying selection maintaining ancestral function, permitting evolution of novel enzymatic properties optimized for prey subjugation rather than cellular maintenance. The accelerated evolution evident in venom gene sequences, with substitution rates sometimes exceeding those in neutral sequences, indicates strong positive selection driving functional diversification in response to prey resistance evolution or shifts in dietary preferences that favor alternative toxin profiles. Regulatory evolution also contributes to venom variation, with expression level changes altering toxin ratios in venom cocktails without requiring structural changes in toxin molecules themselves, providing an additional axis of variation that can respond to selection on venom phenotypes. Forensic applications of snake venom analysis have emerged as molecular techniques enable identification of venom proteins in biological samples, potentially permitting species-level identification of snakes responsible for envenomation when animals are not captured or observed. The proteomic profiles of venoms exhibit species-specific variation that can serve as molecular fingerprints, though the interpretation of venom protein data from clinical samples remains complicated by individual variation in venom composition, degradation of proteins in biological tissues, and analytical challenges in detecting proteins present in low concentrations. Geographic variation in venom composition within species creates additional complexity, as populations separated by substantial distances may exhibit venom profiles as divergent as those distinguishing closely related species, potentially confounding identification efforts that assume geographically homogeneous venom phenotypes. Despite these challenges, forensic venomic approaches show promise for addressing envenomation cases where species identification based on victim descriptions proves unreliable or impossible, potentially improving treatment outcomes through more accurate matching of patients with appropriate antivenoms. The microbial communities inhabiting snake oral cavities and venom glands represent poorly explored frontiers in snake biology, potentially harboring novel antimicrobial compounds or contributing to envenomation pathology through introduction of bacteria into wounds during bites. Recent culture-independent surveys employing high-throughput DNA sequencing have revealed diverse bacterial and fungal assemblages in venom and oral samples, though the functional roles of these microorganisms remain largely speculative. Some researchers hypothesize that oral bacteria contribute to prey mortality through sepsis following envenomation, effectively functioning as biological enhancers of venom toxicity, though controlled studies are required to evaluate whether bacterial introduction significantly affects prey mortality rates beyond effects attributable to venom alone. Alternative hypotheses suggest that microorganisms produce antimicrobial compounds that protect snakes from infections following feeding on potentially contaminated prey, or that microbial communities in venom glands neutralize toxic secondary metabolites in prey tissues that might otherwise harm snakes consuming chemically defended prey. Telomere dynamics in snakes offer intriguing insights into aging biology, with some species exhibiting negligible senescence and maintaining telomere lengths throughout life in contrast to the telomere attrition characteristic of aging in many mammals and birds. The mechanisms underlying telomere maintenance in snakes remain incompletely characterized, though constitutive expression of telomerase, the enzyme that adds telomeric repeats to chromosome ends, likely contributes to the lack of replicative aging observed at the cellular level in some species. The ecological and evolutionary implications of negligible senescence include potentially indeterminate growth, where individuals continue increasing in size throughout life rather than asymptoting at mature size, creating populations where age structure correlates strongly with size structure, and the largest individuals represent the oldest members of populations. The reproductive output of indeterminately growing females typically scales with body size, creating positive correlations between female age and fecundity that contrast with the reproductive senescence characteristic of many other vertebrates, potentially favoring iteroparity and long lifespans when adult survival rates remain high. Parthenogenesis, or reproduction without fertilization, occurs facultatively in several snake species, providing reproductive assurance when mates prove unavailable while maintaining the option of sexual reproduction when males are present. The chromosomal mechanisms underlying parthenogenesis vary across species, with some employing premeiotic doubling that generates offspring genetically identical to mothers, while others use terminal fusion or central fusion of meiotic products that generate reduced heterozygosity relative to maternal genotypes. The fitness consequences of parthenogenesis include loss of genetic diversity in parthenogenetic lineages and expression of deleterious recessive alleles that would remain masked by heterozygosity in sexually produced offspring, though the immediate benefit of producing offspring versus remaining nulliparous may outweigh these long-term genetic costs in situations where mate encounter rates are extremely low. Facultative parthenogenesis has been documented in various captive snakes including pythons, boas, and pit vipers, raising intriguing questions about prevalence of this reproductive mode in wild populations and circumstances triggering its expression. The study of snake fossils provides temporal perspective on modern diversity, revealing extinct lineages that attained tremendous sizes or possessed unique morphological features lost during subsequent extinction events. Titanoboa, discovered in Paleocene deposits of Colombia and dating to approximately 60 million years ago, represents the largest snake known from the fossil record, with estimated lengths approaching 13 meters and body masses exceeding 1,000 kilograms. The existence of such gigantic snakes implies considerably warmer paleoclimates than present, as the thermoregulatory constraints affecting modern snakes would prevent attainment of such massive sizes under current thermal regimes where insufficient environmental heat availability would limit activity budgets. The inference of paleoclimatic conditions from ectotherm body sizes represents an active area of paleobiological research, with theoretical models linking maximum sustainable size to environmental temperature through effects on metabolic scope and activity patterns, though considerable uncertainty surrounds the precise calibration of these relationships for extinct taxa. Snake venom's effects on pain perception have revealed insights into nociceptive signaling pathways, with some toxins specifically targeting ion channels involved in transmitting pain signals, either enhancing pain sensation through direct activation of nociceptors or paradoxically reducing pain through desensitization of these same receptors at higher concentrations. The bidirectional modulation of pain by venom components has attracted pharmaceutical interest as potential leads for analgesic drug development, with particular focus on toxins targeting transient receptor potential channels and voltage-gated sodium channels that play crucial roles in nociception. The development of pain medications based on venom peptides faces significant challenges, including poor oral bioavailability of large peptide molecules, potential off-target effects, and the need for extensive preclinical testing, though successful examples like ziconotide, a synthetic version of cone snail venom peptide approved for treating severe chronic pain, demonstrate the feasibility of translating venom research into clinical applications that benefit patients suffering from debilitating pain syndromes resistant to conventional analgesics. The ecological cascade effects of snake population declines have begun receiving attention as researchers document changes in prey populations and vegetation structure following snake extirpation or severe population reduction. Mesopredator release, where intermediate-level predators increase in abundance following removal of apex predators that previously controlled their populations, may occur when snake populations decline substantially, permitting smaller predators that snakes formerly consumed to achieve higher densities, potentially increasing predation pressure on shared prey or exploiting prey unavailable to snakes. The resulting shifts in food web structure can propagate through multiple trophic levels, affecting vegetation communities through altered herbivore pressure or modifying competitive dynamics among prey species that experience differential predation from released mesopredators versus snakes. The documentation and quantification of these ecological cascades remain challenging due to the multiplicity of factors simultaneously affecting ecosystems and the difficulty of experimentally manipulating snake populations at scales relevant to ecosystem processes, though comparative studies of systems with and without snakes provide suggestive evidence for their regulatory importance. The global distribution of snakebite incidence exhibits strong geographic patterns, with tropical and subtropical regions of Africa, Asia, and Latin America bearing disproportionate burdens of envenomation mortality and morbidity relative to their populations. The social determinants of snakebite risk include agricultural occupations that require fieldwork in snake habitat, housing conditions that permit snake entry into living spaces, limited footwear use, and inadequate access to medical care including antivenom supplies. The recognition of snakebite as a neglected tropical disease by the World Health Organization has catalyzed renewed attention and funding for antivenom development, epidemiological surveillance, and public health interventions targeting high-risk populations, though substantial gaps remain in antivenom availability and affordability in many affected regions. The market economics of antivenom production create perverse incentives where populations most affected by snakebite possess limited purchasing power, reducing commercial incentives for pharmaceutical companies to develop and manufacture region-specific antivenoms despite clear medical needs, requiring public sector intervention and international cooperation to ensure access to these life-saving biologics. Biomechanical studies of snake swimming reveal sophisticated control of body waves that generate thrust while minimizing lateral recoil that would reduce forward progression efficiency. Aquatic snakes modulate wavelength, amplitude, and frequency of body undulations depending on swimming speed and behavioral context, with different gaits employed during routine swimming versus burst swimming when fleeing predators or pursuing agile prey. The added mass effects of water, which effectively increases the inertia that must be accelerated during each tail beat, requires greater force production compared to terrestrial locomotion, with predicted energetic costs of aquatic locomotion exceeding terrestrial movement costs at equivalent speeds. Nonetheless, many snake species readily enter water and demonstrate considerable swimming proficiency, suggesting that the biomechanical and energetic challenges of aquatic movement remain surmountable without specialized morphological adaptations. Though fully aquatic species like sea snakes exhibit morphological modifications including laterally compressed bodies and paddle tails that enhance swimming performance beyond capabilities of terrestrial species. The phenomenon of snake aggregations, where numerous individuals congregate in restricted areas despite generally solitary lifestyles, has attracted considerable research interest focused on understanding proximate mechanisms and ultimate functions underlying these gatherings. Aggregation sites may provide thermal benefits, as mentioned previously regarding hibernacula, or may represent spatial concentrations around limited resources including water sources, prey concentrations, or reproductive opportunities. Some evidence suggests chemical communication via pheromones coordinates aggregation formation and maintenance, with individuals following scent trails deposited by conspecifics to locate aggregation sites, though the sensory mechanisms permitting trail following over substantial distances remain incompletely characterized. The potential costs of aggregation include increased conspicuousness to predators, heightened parasite transmission, and resource competition, creating selective pressures favoring aggregation dissolution once benefits are realized or costs begin exceeding benefits, generating temporally dynamic aggregation patterns that reflect changing cost-benefit ratios across seasons and reproductive states. The impact of introduced snakes on island ecosystems has generated dramatic examples of invasive species effects, most notoriously the brown tree snake introduction to Guam following World War II military activities. The subsequent population explosion of brown tree snakes resulted in catastrophic impacts on native avifauna, driving multiple endemic bird species to extinction or severe population depression through nest predation and predation on adults in what represents one of the most severe documented cases of invasive predator impacts on insular vertebrate communities. The absence of native snake predators and the naivety of island prey to snake predation likely contributed to the severity of impacts, permitting brown tree snakes to achieve extraordinarily high densities unsustainable on continental landmasses where prey populations possess evolutionary experience with snake predation. Control efforts employing various strategies including trapping, toxicant deployment, and barrier construction have achieved limited success in containing brown tree snake populations, highlighting the difficulty of eradicating established invasive reptile populations once they achieve large population sizes across extensive geographic areas. The future of snake biology research promises continued discoveries as new technologies permit previously impossible investigations into physiology, behavior, and ecology. High-resolution tracking devices small enough for implantation in relatively modest-sized snakes enable detailed documentation of movement patterns, habitat use, and thermal biology across seasons, generating behavioral data that inform conservation planning and habitat management. Genomic tools permit population genetic analyses revealing connectivity patterns, identifying conservation units requiring distinct management approaches, and detecting genetic consequences of habitat fragmentation or population declines. Proteomics and transcriptomics applied to venom research continue revealing novel toxins and providing insights into venom evolution, while physiological studies employing increasingly sophisticated instrumentation document performance capabilities and constraints shaping snake ecology and evolution. The integration of these technological advances with traditional natural history observation and experimentation positions snake biology as a dynamic field poised for continued growth and discovery as researchers probe the many remaining mysteries surrounding these remarkable reptiles. The intersection of climate science and snake conservation creates urgent imperatives for understanding thermal biology and predicting distributional responses to warming temperatures. Species distribution models incorporating thermal physiology, dispersal capabilities, and habitat requirements attempt to forecast range shifts under various climate scenarios, though model predictions remain uncertain due to incomplete physiological data for most species and challenges in predicting landscape-level changes in habitat characteristics. Northern range expansions may occur for some temperate species as formerly inhospitable thermal regimes ameliorate, potentially bringing venomous species into human-populated areas previously free of snakebite risk, creating public health concerns requiring proactive planning and education. Conversely, tropical species already operating near their upper thermal tolerances may face contracting suitable habitat as temperatures exceed physiological thresholds, potentially driving extinctions of range-restricted species unable to disperse to cooler refugia or adapt sufficiently rapidly to track shifting climatic envelopes. Human cultural evolution has generated diverse relationships with snakes that transcend simple fear or revulsion, encompassing practices ranging from snake charming to ritual snake handling in religious contexts. The snake charmers of South Asia, Declining in number due to wildlife protection laws and changing cultural attitudes, historically maintained complex relationships with cobras captured from wild populations and induced to rear and hood in response to movements and vibrations rather than actual music, as snakes lack external ears and cannot hear airborne sounds. Religious snake handling practices in some Pentecostal traditions of Appalachia involve handling venomous pit vipers during religious services as demonstrations of faith, practices that occasionally result in envenomation and fatality but persist due to deeply held religious beliefs about divine protection. These diverse cultural practices, while controversial from conservation and safety perspectives, demonstrate the powerful symbolic meanings humans project onto snakes, reflecting our enduring fascination with these limbless predators that simultaneously attract and repel, inspire reverence and terror, and ultimately compel attention through their radical difference from the human form. The sensory world of snakes extends into realms barely comprehensible to humans, with their chemosensory capabilities permitting detection of molecular traces at concentrations measured in parts per trillion, effectively allowing them to perceive chemical landscapes invisible to organisms lacking comparable olfactory sophistication. The paired vomeronasal organs process pheromonal information that guides mate selection, territorial behavior, and prey trail following across distances that can span hundreds of meters in some species. Research employing radiotelemetry has documented male snakes following female pheromone trails over tortuous routes exceeding 1 kilometer, maintaining accurate course despite numerous potential confounding chemical signals from other organisms sharing the habitat. The molecular specificity of vomeronasal receptors permits discrimination between closely related chemical variants, allowing males to assess female reproductive condition, genetic compatibility, and individual identity through pheromone analysis alone. This chemical communication system operates largely outside human perception, creating a parallel sensory universe where information flows through molecular channels rather than the visual and acoustic modalities dominating human experience, reminding us that reality comprises multiple sensory dimensions accessible only to organisms possessing appropriate receptor systems. The ontogeny of venom production in neonate snakes raises fascinating questions about the timing and regulation of toxin synthesis during development. Studies examining venom gland development have revealed that functional venom-producing cells differentiate during late embryonic stages, permitting some species to possess fully functional venom systems at birth or hatching. The composition of neonate venom often differs substantially from adult venom in the same species, typically containing higher concentrations of neurotoxic components relative to tissue-destructive enzymes, a pattern potentially reflecting the smaller prey items neonates consume and the reduced mechanical damage smaller prey require for subjugation. Alternative hypotheses suggest that neonate venom composition optimizes defensive rather than predatory functions, as newly independent young face intense predation pressure and may employ venom primarily for predator deterrence rather than prey capture. The developmental regulation of venom gene expression remains incompletely understood, though transcriptomic studies have identified regulatory networks controlling the temporal activation of toxin genes during gland maturation, suggesting sophisticated developmental programs coordinate venom system ontogeny with broader growth trajectories. The phylogenetic distribution of venom across snake diversity reveals multiple independent origins of venom systems, challenging earlier assumptions that venom evolved once in snake evolutionary history and subsequently diversified through modification of ancestral toxins. Comparative genomic and transcriptomic analyses have identified venom-like genes in species traditionally considered non-venomous, suggesting that the genetic toolkit for venom production may be more widespread than delivery mechanisms permitting effective envenomation. This discovery implies that the distinction between venomous and non-venomous snakes may reflect variations in delivery efficiency and toxin potency rather than fundamental differences in the presence or absence of toxin-encoding genes. The rear-fanged colubrid snakes, long dismissed as harmlessly non-venomous, include species capable of delivering medically significant envenomations through enlarged posterior maxillary teeth and modified salivary secretions containing toxic proteins. Several human fatalities attributed to rear-fang species, including the boneslong and twig snakes of Africa, demonstrate that effective venom delivery need not require the sophisticated front-fanged mechanisms characteristic of vipers and elapids, as sustained chewing can introduce sufficient toxin quantities to produce life-threatening effects. Hybrid zones where distinct snake species or subspecies interbreed provide natural laboratories for studying reproductive isolation mechanisms and the genetic architecture of species boundaries. The extensive hybridization documented between certain rattlesnake species in regions of range overlap generates admixed populations exhibiting intermediate phenotypes and novel trait combinations absent in parental populations. The fitness consequences of hybridization remain debated, with some studies suggesting hybrid individuals exhibit reduced survival or reproductive success compared to pure parental types, indicating partial reproductive isolation maintained by selection against hybrids. Conversely, other systems show little evidence for hybrid disadvantage, with admixed populations persisting across multiple generations without trending toward one parental type, suggesting that reproductive barriers remain incomplete and gene flow continues despite species-level taxonomic distinctions. The genomic analysis of hybrid zones permits identification of genomic regions showing reduced introgression, likely harboring genes contributing to reproductive isolation or local adaptation, while highly introgressed regions presumably contain loci where allelic variants function equivalently across genetic backgrounds, permitting unrestricted gene flow at those locations. The muscular hydrostat mechanics underlying tongue projection in snakes represents a remarkable example of how animals lacking rigid skeletal supports can generate rapid, forceful movements through manipulation of incompressible tissue volumes. The tongue muscles, arranged in orthogonal orientations, permit precisely controlled shape changes that extend the tongue from the oral cavity during chemosensory sampling. The rapidity of tongue flicking, which can occur multiple times per second in some species, requires sophisticated neural control coordinating muscle activations with respiratory cycles, as tongue projection must occur during specific respiratory phases to avoid interference with breathing. The tongue's forked morphology, mentioned earlier regarding its chemosensory function, also provides aerodynamic benefits that reduce air resistance during high-speed projection, permitting faster flicking rates with reduced energetic costs. Comparative studies examining tongue morphology across snake phylogeny have revealed correlated evolution between tongue structure and chemosensory organ anatomy, suggesting coevolutionary refinement of these integrated systems over millions of years as selection favored enhanced chemical detection capabilities. Snake scales represent modified epidermal structures rather than dermal ossifications like fish scales, developing through regional specialization of skin epithelium into overlapping protective units that provide flexibility while resisting abrasion and desiccation. The microscopic structure of scales varies across body regions and between species, reflecting diverse functional demands including locomotion, defense, and water retention. Ventral scales in terrestrial species are typically enlarged and possess enhanced keratinization compared to dorsal scales, providing durable surfaces that withstand repeated substrate contact during locomotion. The process of ecdysis, or skin shedding, occurs periodically throughout snake lifetimes as worn outer skin layers are replaced by newly formed underlying epidermis. The shedding cycle begins with separation of old epidermis from newly forming layers beneath, creating a cloudy appearance as lymphatic fluid accumulates in the separation space, temporarily obscuring vision as the spectacle over each eye becomes opaque. Once separation completes, snakes typically initiate shedding by rubbing their snouts against rough substrates to create initial tears in the old skin, then crawling forward to peel the shed skin inside out as it catches on environmental irregularities. The frequency of shedding varies with growth rate, nutritional status, and reproductive condition, with rapidly growing juveniles shedding as frequently as monthly while growth-limited adults may shed only 2 or 3 times annually. Reproductive females may shed immediately before egg deposition or parturition, possibly reflecting hormonal influences on skin turnover or preparation for the post-reproductive growth phase. Unsuccessful sheds, where patches of old skin remain adhered to the body, can indicate underlying health problems including dehydration, malnutrition, parasitic infections, or inadequate environmental humidity. Retained spectacles over the eyes pose particular concerns as accumulating layers can impair vision progressively across multiple incomplete sheds, potentially compromising hunting success and predator detection. Captive husbandry protocols emphasize providing appropriate humidity and textured surfaces facilitating complete sheds, as artificial environments may lack the moisture gradients and substrate variability that wild snakes exploit during natural shedding cycles. The evolution of different fang types reflects diverse solutions to venom delivery challenges, with solid rear fangs, grooved rear fangs, fixed front fangs, and hinged front fangs each offering distinct advantages and constraints. Solid rear fangs provide minimal venom delivery capacity, requiring prolonged chewing to introduce toxins through tooth punctures, yet their simple construction requires minimal morphological specialization and permits retention of normal feeding tooth functions. Grooved fangs channel venom along external grooves through capillary action, improving delivery efficiency compared to solid teeth while maintaining structural simplicity. Fixed front fangs, characteristic of elipid snakes including cobras and krites, provide excellent venom delivery through hollow tubes connecting venom glands to fang tips, though their fixed erect position constrains maximum fang length since excessively long fangs would prevent mouth closure or pierce the snake's own lower jaw. The hinged fangs of vipers solve this constraint through a rotating maxillary bone that permits extraordinarily long fangs to fold against the palate when the mouth closes, then swing forward during strikes to project anteriorly like biological switchblades, maximizing venom injection depth and permitting strikes with minimal mouth opening. The immune responses generated following envenomation represent complex interactions between venom components and victim immune systems, sometimes producing effects as dangerous as the direct toxin actions themselves. Anaphylactic reactions to venom proteins can occur in previously sensitized individuals, generating life-threatening systemic allergic responses characterized by airway constriction, cardiovascular collapse, and widespread mast cell degranulation releasing inflammatory mediators. The risk of anaphylaxis complicates antivenom therapy, as antivenoms themselves, being derived from horse or sheep serum containing foreign proteins, can trigger allergic reactions including potentially fatal anaphylactic shock. Modern antivenom production employs various purification techniques to remove non-antibody proteins from therapeutic preparations, reducing but not eliminating allergic reaction risks. Serum sickness, a delayed immune reaction to foreign proteins characterized by fever, rash, joint pain, and malaise, develops in a substantial minority of antivenom recipients days to weeks after treatment, reflecting immune complex formation and tissue deposition. These immunological complications necessitate careful clinical monitoring of envenomation victims even after apparent initial recovery, as delayed reactions may require additional medical intervention. The genetic basis of venom resistance in prey populations has become increasingly tractable through molecular approaches permitting identification of specific mutations conferring reduced toxin sensitivity. Studies of venom-resistant ground squirrels, possums, and hedgehogs have revealed multiple independent origins of resistance through convergent molecular evolution affecting toxin target proteins. Modifications in acetylcholine receptor sequences that reduce neurotoxin binding affinity while preserving normal receptor function exemplify the exquisite evolutionary fine-tuning required, as mutations must reduce toxin binding without compromising neurotransmitter binding or channel function. The population genetics of venom resistance reveal frequency-dependent selection dynamics where resistance alleles reach intermediate frequencies rather than fixing completely, suggesting fitness costs associated with resistance that prevents selective sweeps. These costs might include reduced receptor efficiency, metabolic burdens of producing modified proteins, or pleiotropic effects where resistance mutations affect other physiological processes beyond toxin interactions. The geographic mosaic of resistance allele frequencies correlates with local snake abundance, providing compelling evidence for predator-mediated selection driving resistance evolution in areas of high envenomation risk. Maternal effects in snakes extend beyond simple provision of yolk nutrients, encompassing influences on offspring phenotype through embryonic thermal environments, hormone transfer, and antibody transmission that shape juvenile survival and performance. Temperature-dependent sex determination, documented in several snake species, represents an extreme maternal effect where incubation temperatures during critical developmental periods determine offspring sex ratios, permitting mothers selecting nest sites to potentially manipulate sex ratios toward locally advantageous values. The transfer of maternal antibodies to developing embryos provides passive immunity during vulnerable early life stages when juvenile immune systems remain functionally immature, conferring protection against pathogens the mother encountered and mounted immune responses against during her lifetime. This transgenerational immune protection proves particularly valuable for viviparous species where extended embryonic retention permits substantial antibody transfer, potentially explaining some of the selective advantages favoring viviparity evolution despite the costs of prolonged embryo retention. Maternal stress hormones transferred to developing embryos can program offspring phenotypes toward cautious, stress-reactive behavioral profiles that may prove adaptive in dangerous environments but incur costs in benign conditions, creating environment-dependent fitness consequences of maternal effects. The biomechanics of prey constriction employed by boids and pythons involves sophisticated force regulation where snakes sense prey respiratory movements and circulatory pressure, modulating constriction pressure to maintain sufficient force for circulatory disruption without expending excessive energy on already incapacitated prey. Recent studies have overturned long-held assumptions that constriction kills through asphyxiation, demonstrating instead that constrictors induce immediate circulatory collapse through compression of major vessels and the heart itself, producing rapid unconsciousness through cerebral ischemia. The pressure sensors distributed along constrictor bodies detect minute thoracic movements indicating continued breathing attempts or heartbeats, triggering additional coil tightening that prevents recovery while the snake waits for all physiological activity to cease. This sensory feedback system permits efficient prey dispatch using minimum necessary force, as excessive constriction wastes energy while insufficient pressure allows prey to potentially escape. The transition from active constriction to relaxation following prey death reflects sophisticated sensory motor integration that permits constrictors to accurately assess when prey has succumbed, minimizing handling time before swallowing can commence. Arboreality in snakes correlates with numerous morphological and physiological adaptations beyond those mentioned earlier, including cardiovascular modifications preventing orthostatic hypotension when individuals position their heads above their hearts during vertical climbing. The gravitational pressure gradients opposing blood return to the heart from elevated body regions would induce consciousness-threatening cerebral hypoperfusion without compensatory mechanisms including enhanced venous tone and cardiac contractility. Studies measuring blood pressure at different body positions in arboreal versus terrestrial species have documented substantially higher baseline blood pressures in arboreal forms, providing the additional pressure head required to perfuse the brain against gravitational opposition. These cardiovascular adaptations carry potential costs including increased cardiac workload and elevated stroke risk from chronic hypertension, suggesting that arboreality imposes physiological trade-offs that constrain which species can successfully invade canopy environments. The evolution of arboreality has occurred independently numerous times across snake phylogeny, with convergent evolution of slender body forms, prehensile tails, and cardiovascular modifications reflecting predictable selective responses to common challenges posed by three-dimensional arboreal environments. Communal nesting, where multiple females deposit eggs in shared locations, occurs in various snake species and may confer benefits including reduced individual predation risk through dilution effects, improved nest microclimate through combined metabolic heat production from developing embryos, and reduced search costs for suitable nest sites in landscapes where appropriate locations are spatially limited. The potential costs of communal nesting include increased conspicuousness to nest predators, enhanced pathogen transmission among embryos sharing confined spaces, and possible resource depletion if nest sites provide limited critical resources. The evolution of communal nesting may be driven more by limited availability of suitable nest sites than by active benefits of aggregation, with site limitation forcing females to accept suboptimal social conditions as preferable to utilizing thermally or hydrically unsuitable solitary sites. Genetic analyses of hatchlings from communal nests have revealed multiple maternity and sometimes multiple paternity within nests, indicating that communal nesting does not necessarily imply cooperative breeding but rather represents aggregation of independently reproducing females exploiting shared resources. The maintenance of these aggregations across generations suggests that benefits outweigh costs sufficiently for natural selection to favor nest site fidelity and tolerance of conspecific females during the vulnerable egg-laying period. The coloration patterns adorning snakes serve multiple functions including camouflage, aposematism, thermoregulation, and possibly intraspecific communication, with pattern diversity across species reflecting varied selective pressures operating in different ecological contexts. Countershading, where dorsal surfaces are darker than ventral surfaces, creates visual illusions that flatten three-dimensional appearance through compensating for typical lighting conditions where illumination arrives predominantly from above. Disruptive patterns featuring high-contrast patches break up body outlines, making stationary snakes harder to detect against complex backgrounds despite imperfect background matching. Ontogenetic color changes occurring as juveniles mature into adults may reflect shifting ecological roles, with juvenile patterns optimizing camouflage against different backgrounds than adults occupy or signaling subordinate status to conspecifics, reducing aggression from territory-holding adults. The genetic and developmental mechanisms controlling color pattern formation remain active research areas, with recent studies identifying regulatory genes controlling melanin synthesis and distribution that underlie pattern variation within and between species, providing insights into how evolutionary modifications of development generate phenotypic diversity. Sexual dimorphism in snakes, though less dramatic than in many other vertebrates, includes consistent differences in body size, tail length, and head proportions between sexes within species. Females typically exceed males in body length in most species, reflecting fecundity advantages of large body size as larger females can allocate more resources to egg or embryo production. Males often possess relatively longer tails than females, a pattern reflecting the presence of paired hemipenes inverted within the tail base that require additional caudal length to accommodate these copulatory organs. Head size sexual dimorphism, particularly pronounced in some vipers and water snakes, may reflect sexual selection on male compatibility where larger-headed males achieve dominance and preferential mating access, or could indicate dietary niche partitioning between sexes reducing intraspecific competition. The hormonal control of sexually dimorphic traits involves sex steroid hormones including testosterone and estrogen that coordinate expression of secondary sexual characteristics with gonadal maturation, ensuring that sex-specific phenotypes develop appropriate to reproductive roles. Experimental hormone manipulations have demonstrated that many sexually dimorphic traits remain phenotypically plastic, developing differently depending on hormonal environments during critical developmental windows, illustrating the importance of endocrine signaling in orchestrating sexual differentiation. Tail autonomy, though less common in snakes than lizards, occurs in several lineages and represents a last resort defense permitting escape from predator grasp at the cost of permanent tail loss since snakes cannot regenerate lost caudal segments. The breaking typically occurs at predetermined fracture planes between vertebrae where specialized connective tissue permits rapid separation when muscles contract forcefully, generating shearing forces exceeding tissue strength. The autotomized tail segment may continue moving for several minutes after separation, potentially distracting predators and permitting the snake to escape during this window. The costs of tail loss include reduced locomotor performance, lost energy storage in caudal fat reserves, and impaired reproductive capability in males whose hemipenes become inaccessible if tail loss occurs proximally enough to remove reproductive structures. Despite these costs, the prevalence of tail loss scars in some wild populations suggests that autotomy successfully facilitates predator escape with sufficient frequency to maintain the trait despite its disadvantages. The evolution of tail autotomy likely reflects strong predation pressure favoring any escape mechanism, with tail loss representing a costly but preferable alternative to death in predator-prey encounters where other defenses prove insufficient. Phenotypic plasticity in snakes permits individuals to adjust their phenotypes in response to environmental conditions, generating variation within genotypes that may enhance fitness in heterogeneous or unpredictable environments. Temperature-dependent developmental plasticity affects numerous traits including growth rates, adult body sizes, and behavioral temperaments, with embryos developing at cooler temperatures typically emerging at smaller sizes but sometimes exhibiting compensatory growth that eventually eliminates size differences. Dietary plasticity allows snakes to adjust digestive physiology and gut morphology to match prey characteristics, with individuals consistently feeding on large meals developing more massive digestive systems than conspecifics consuming smaller, more frequent meals. This digestive plasticity extends to biochemical levels, with enzyme production and secretion rates matching substrate availability, illustrating organism-environment feedback where phenotypes track ecological conditions rather than remaining fixed by genetic programs alone. The extent of plasticity varies across traits and species, with some characteristics exhibiting substantial environmental sensitivity while others remain canalized despite environmental variation, reflecting different evolutionary histories and functional requirements constraining acceptable variation ranges. Snake navigation during long-distance movements, including migrations between summer foraging areas and winter hibernacula or between terrestrial and aquatic habitats in semi-aquatic species, likely integrates multiple sensory modalities including solar compass orientation, magnetic compass sensitivity, and landmark recognition. Experimental studies have demonstrated that some snake species can orient homeward after displacement from capture sites, though orientation success varies with displacement distance and the familiarity of release sites. The sensory basis of snake navigation remains incompletely understood, though evidence suggests chemosensory trail following contributes substantially to relocating previously visited locations where scent marks provide directional information. The cognitive capabilities required for successful navigation, including spatial memory formation and maintenance, suggest more sophisticated neural processing than often attributed to reptilian brains, challenging outdated assumptions about reptile cognitive limitations. Recent studies employing GPS tracking have documented remarkably straight-line movements over multi-kilometer distances in some species, suggesting genuine navigation using distant cues rather than random searching or simple kinesis responses to local gradients, though the specific cues employed remain uncertain and may vary across species and contexts. Parasites afflicting snakes include diverse taxa spanning protozoans, helminths, and arthropods that exploit serpentine hosts through various transmission pathways and cause morbidity ranging from negligible to fatal depending on parasite species, infection intensity, and host condition. Hemogregarines, protozoan blood parasites transmitted by blood-feeding mites and mosquitoes, infect numerous snake species but typically cause minimal pathology unless infections reach very high intensities overwhelming immune defenses. Nematode parasites inhabiting respiratory tracts, digestive systems, or body cavities can induce severe disease through physical blockage, tissue destruction, or nutrient competition, particularly when environmental stressors compromise host immune function permitting parasite proliferation. Ticks and mites parasitizing snake skin cause irritation, blood loss, and pathogen transmission, with heavy infestations potentially inducing anemia or secondary bacterial infections where mouthpart attachment sites breach integumentary barriers. The coevolutionary dynamics between snakes and their parasites generate ongoing arms races where hosts evolve enhanced immune defenses while parasites evolve immune evasion mechanisms, creating cycles of adaptation and counteradaptation that maintain parasite diversity and infection prevalence across host populations. Climate-driven range shifts in snake distributions are already documented in some well-studied systems, with northern range boundaries advancing poleward as warming temperatures render previously inhospitable regions thermally suitable for snake survival and reproduction. The rates of range expansion vary considerably across species, likely reflecting differences in dispersal capabilities, habitat specificity, and reproductive rates that constrain how rapidly populations can colonize newly suitable areas. Leading-edge populations expanding into novel regions may encounter altered biotic communities lacking co-evolved predators, competitors, or prey species, creating ecological novelty that could favor rapid evolutionary change as natural selection acts on populations experiencing unprecedented selective environments. Conversely, trailing-edge populations at southern range boundaries may experience increasing heat stress and declining habitat suitability, potentially driving local extinctions that fragment species distributions and isolate populations into disjunct refugia. The conservation implications of these distributional dynamics include urgent needs for protecting climate corridors, facilitating range shifts, and managing source populations that may seed colonization of newly suitable regions as climate envelopes migrate across landscapes. Endoparasites, including pentastomid tongue worms, inhabit respiratory passages of various snake species, attaching to tracheal and lung surfaces where they feed on blood and epithelial tissues while causing inflammation and potentially compromising respiratory function. These bizarre parasites, whose arthropod affinities remained uncertain until molecular phylogenetics clarified their systematic placement, possess highly modified body forms adapted for endoparasitic lifestyles, with anterior hooks facilitating attachment and worm-like body shapes permitting movement through host airways. Life cycles involve intermediate hosts, typically mammals or other vertebrates consumed by snakes, with infective stages penetrating intestinal walls and migrating to respiratory tissues where they mature and reproduce. The specificity of pentastomid host associations varies, with some parasite species exhibiting strong host specificity while others infect multiple snake species, reflecting different evolutionary histories and ecological opportunities for host switching. The pathological impacts range from minimal in light infections to severe respiratory compromise in heavy infestations, particularly when multiple parasites obstruct airflow or secondary bacterial infections colonize damaged respiratory epithelium. Snake communities in diverse tropical ecosystems can comprise over 50 sympatric species occupying distinct ecological niches defined by microhabitat preferences, activity periods, and dietary specializations that reduce interspecific competition. The mechanisms permitting such high diversity include fine-scale habitat partitioning where species segregate vertically into ground, understory, and canopy layers, temporal partitioning where diurnal, crepuscular, and nocturnal activity patterns reduce encounter rates between competitors, and dietary partitioning where species specialize on different prey taxa or size classes. The coexistence of multiple potentially competing species challenges classical competitive exclusion principles, suggesting that environmental heterogeneity, disturbance regimes, and predation pressure may prevent competitive exclusion by creating shifting mosaics of conditions where no single species can monopolize resources across all spatial and temporal scales. The study of snake community structure has illuminated general principles of community assembly and coexistence, demonstrating that high local diversity can emerge through accumulation of specialists rather than dominance by superior competitors that might exclude subordinate species in less heterogeneous environments. Fossil snakes occasionally preserve exceptional details including skin impressions, gut contents, and even color patterns visible through preservation of melanosomes, the cellular organelles containing pigments responsible for integumentary coloration. These rare exceptional fossils provide unprecedented insights into the paleobiology of extinct species, revealing dietary preferences through identifiable prey remains, documenting ancient color patterns suggesting cryptic or aposematic strategies similar to modern species, and demonstrating that fundamental aspects of snake biology remained consistent across tens of millions of years despite substantial taxonomic turnover. The taphonomic biases affecting snake fossil preservation, particularly the fragility of snake skeletons and their susceptibility to disarticulation, mean that most fossil snakes are represented by fragmentary remains offering limited insight into whole organism biology. Consequently, each exceptional fossil revealing soft tissue details or behavioral information represents an invaluable data point constraining reconstructions of ancient ecosystems and snake evolution, though the rarity of such specimens leaves substantial gaps in our understanding of snake paleobiology. The economic value of snake skins in leather industries creates market demand driving legal and illegal harvest that threatens some populations, particularly large-bodied species whose skins command premium prices in fashion markets. The international trade in python and boa skins involves millions of individual animals annually, with major exporting countries including Southeast Asian nations and some African countries where regulatory oversight may prove inadequate to ensure sustainable harvest rates. The implementation of CITES regulations governing international trade in endangered species provides frameworks for monitoring and regulating trade, though enforcement challenges and illegal trafficking continue undermining conservation efforts. Sustainable leather production from farmed snakes offers potential alternatives to wild harvest, though market preferences for wild-caught skins and higher production costs associated with captive breeding create economic incentives favoring continued wild harvest despite its conservation risks. Consumer education highlighting the conservation impacts of exotic leather purchases could reduce demand for wild-caught products, shifting market preferences towards sustainably sourced alternatives, though changing entrenched consumer preferences faces significant challenges. Snake venoms contain compounds exhibiting antimicrobial activities that potentially protect snakes from infections that could arise following feeding on bacteria-laden prey or after sustaining injuries during prey capture or conspecific combat. The antimicrobial peptides isolated from various snake venoms show activity against diverse bacterial and fungal pathogens, suggesting that venoms serve dual functions as offensive predatory weapons and defensive antimicrobial arsenals. The therapeutic potential of these antimicrobial compounds has attracted pharmaceutical interest as antibiotic resistance grows increasingly problematic, with venom-derived antimicrobials offering novel molecular scaffolds distinct from conventional antibiotics that might overcome existing resistance mechanisms. The development of antimicrobial drugs from venom peptides faces challenges including potential toxicity to host tissues, difficulty synthesizing or producing compounds at commercial scales, and the extensive regulatory testing required before human therapeutic use. Though successful development would validate venoms as sources of clinically valuable drugs beyond the cardiovascular and pain management applications already achieved. The role of snakes as umbrella species in conservation planning recognizes that protecting habitat sufficient for maintaining viable snake populations likely benefits numerous co-occurring species sharing similar habitat requirements. The large home ranges and specific microhabitat needs of some snake species mean that reserve designs accommodating their spatial requirements will encompass substantial habitat areas supporting diverse assemblages of plants and animals. The use of snakes as flagship species in conservation campaigns remains more challenging than using charismatic megafauna, given widespread negative perceptions, though education emphasizing ecological roles and intrinsic value has achieved some success in shifting public attitudes. The integration of snake conservation into broader ecosystem management frameworks ensures that these often overlooked predators receive consideration in land use planning, habitat restoration, and protected area management, potentially preventing declines that might otherwise occur through benign neglect or active persecution. The monitoring of snake populations as indicators of ecosystem health provides early warning signals of environmental degradation, as declines often precede more obvious deterioration visible to casual observers, permitting proactive intervention before irreversible damage occurs. Intersexual conflict in snake mating systems emerges when male and female reproductive interests diverge, generating selection for traits in one sex that enhance its fitness at the expense of the other sex. Forced copulation attempts documented in some species reflect male reproductive strategies maximizing mating opportunities regardless of female preferences, potentially imposing costs on females including injury, infection risk, and energetic expenditures resisting unwanted mating. The evolution of female counteradaptations including behavioral resistance, anatomical barriers to insemination, and physiological mechanisms permitting cryptic female choice after copulation represents ongoing arms races between the sexes. Cryptic female choice, where females influence paternity outcomes through post-copulatory mechanisms including differential sperm storage, selective sperm utilization, or facultative abortion of embryos sired by inferior males, provides females with ultimate control over reproductive outcomes despite lacking complete control over mating itself. The documentation of multiple paternity in many snake clutches, where offspring within single reproductive events exhibit multiple sires, suggests that polyandry confers fitness benefits to females that outweigh costs associated with multiple mating, possibly through genetic diversity benefits, fertility assurance, or material benefits obtained from multiple males. Thermoconformity in snakes creates profound temporal heterogeneity in physiological performance, with individuals experiencing dramatic performance variation across daily and seasonal thermal cycles that constrain when various activities can be performed effectively. Hunting success likely varies substantially with body temperature, as strike speed, accuracy, and sensory acuity all exhibit thermal sensitivity, creating time windows when foraging proves most profitable and periods when hunting would likely fail despite energetic investment. Similarly, predator evasion capabilities vary thermally, with cold snakes exhibiting sluggish responses potentially rendering them vulnerable to predators despite possessing adequate escape abilities when warm. This performance variation generates selection for sophisticated thermoregulatory behavior, permitting individuals to maintain body temperatures within preferred ranges during activity periods, though achieving preferred temperatures requires time investments in basking or seeking thermal microenvironments that reduce time available for other fitness-relevant activities. The trade-offs between time spent thermoregulating versus foraging, reproducing, or engaging in other behaviors likely shape optimal activity budgets, with theoretical models predicting how time allocation should vary with environmental temperatures, metabolic requirements, and the thermal sensitivity of different activities. Snake venoms exhibit enormous variation in composition even within species, with individual, sexual, ontogenetic, geographic, and seasonal variation all documented across various taxa. Individual variation in venom composition likely reflects genetic polymorphism at loci encoding toxin proteins, creating standing variation that selection could act upon if environmental conditions favor particular venom phenotypes. Sexual dimorphism in venom composition remains poorly studied but may occur if males and females face different ecological challenges requiring differently optimized venom cocktails, though evidence remains limited and inconsistent across species. Ontogenetic venom variation mentioned earlier reflects developmental programs adjusting toxin expression to match changing ecological roles as snakes grow, potentially minimizing costs of producing toxins inappropriate for current dietary niches. Geographic venom variation within species may reflect local adaptation to regional prey communities exhibiting different susceptibilities to particular toxins, generating geographic mosaics where locally optimized venoms enhance feeding efficiency on local prey but prove less effective against prey populations from different regions. Seasonal venom variation potentially reflects changing metabolic priorities or breeding-related hormonal fluctuations affecting venom gland function, though mechanisms underlying such variation remain speculative and require further investigation. The acoustic environment of snakes remains relatively unexplored, with most species considered deaf to airborne sounds given the absence of external ears and tympanic membranes. However, snakes possess inner ear structures including cochlear apparatus capable of detecting substrate-borne vibrations transmitted through cranial bones, effectively providing seismic rather than aerial acoustic sensitivity. Recent studies have challenged assumptions about snake hearing, demonstrating that some species respond behaviorally to airborne sounds across broader frequency ranges than previously suspected, possibly through skull resonance channeling sound pressure to inner ear structures. The functional significance of this vibroacoustic sensitivity may include detection of approaching predators, recognition of prey movement sounds, or possibly reception of conspecific acoustic signals produced by various behaviors including defensive hissing or tail vibration. The production of sounds by snakes through hissing, which involves forcing air through respiratory passages to generate turbulent flow noise, serves defensive functions by startling or intimidating threats, with variations in hiss characteristics potentially encoding information about snake size or defensive motivation that receivers could theoretically assess and respond to appropriately. Climate change impacts on snake phenology extend beyond simple advances in spring emergence, encompassing alterations in reproduction timing, shifts in brumation entry dates, and mismatches between snake and prey phenologies that could reduce foraging success if temporal overlap between snakes and prey declines. The plastic responses of snakes to changing thermal regimes may buffer some climate impacts if individuals adjust behaviors and physiologies appropriately for current conditions, though the limits of plasticity and the extent to which rapid climate change may exceed plastic response capacities remain uncertain. Evolutionary adaptation to changing conditions represents another potential response pathway, particularly for species with short generation times permitting rapid evolution, though many long-lived snake species with delayed maturity may experience insufficient generations for adaptive evolution to track rapidly shifting environments. The synergistic interactions between climate change and other anthropogenic stressors, including habitat loss and fragmentation, create cumulative impacts potentially exceeding the sum of individual stressors, as populations already stressed by habitat degradation may lack the demographic resilience to additionally cope with climate-driven challenges, driving accelerated declines toward extinction thresholds. The microbiomes inhabiting snake digestive tracts remain poorly characterized relative to mammalian gut microbiomes, though preliminary studies suggest lower bacterial diversity than mammals, possibly reflecting the intermittent feeding typical of snakes and the extreme pH fluctuations occurring between feeding and fasting states. The functional roles of gut microbiota in snake digestion remain speculative, though possibilities include supplementary digestive enzyme production, synthesis of essential nutrients including vitamins, and immune system education similar to microbiome functions documented in other vertebrates. The effects of antibiotics on snake health and physiology remain virtually unstudied, though microbiome disruption could potentially impair digestive efficiency or immune function if microbial communities provide functions critical for host health. The transmission of microbiomes between snake generations likely occurs through environmental acquisition rather than vertical transfer from parents to offspring, given that oviparous species lack physical contact between generations, though viviparous species might permit some maternal microbiome transmission to developing young. Captive snakes likely harbor altered microbiomes compared to wild conspecifics, given different dietary regimes and environmental conditions, potentially affecting health and disease susceptibility in ways not yet recognized or studied systematically. Sperm storage in female snakes permits temporal decoupling of copulation from ovulation and fertilization, with females of various species documented retaining viable sperm for months or even years before utilizing stored sperm for fertilization. The anatomical sites of sperm storage typically include specialized regions of the oviduct featuring tubular glands or crypts where sperm reside protected from female immune responses while maintaining fertilization competence. The physiological mechanisms maintaining sperm viability during prolonged storage remain incompletely understood but likely involve provision of nutrients, removal of metabolic wastes, and protection from oxidative damage that would otherwise degrade sperm function over time. The adaptive significance of sperm storage includes facilitating reproduction in situations where mate encounter rates are low or temporal separation between optimal mating periods and optimal embryonic development periods favors delaying fertilization until environmental conditions suit offspring production. Additionally, sperm storage from multiple males permits post-copulatory sexual selection through sperm competition or cryptic female choice, generating opportunities for females to bias paternity toward preferred males even after copulating with multiple partners. The relative contributions of sperm competition versus female choice in determining paternity patterns remain debated and likely vary across species depending on the specific mechanisms operating and the extent of female control over stored sperm populations. The sensory basis of prey detection in snakes integrates multiple modalities operating over different spatial scales, with long-range detection potentially employing chemosensory cues carried on air currents, intermediate-range detection utilizing visual or thermal cues, and close-range assessment employing tactile and gustatory senses during prey handling and consumption decisions. The relative importance of different sensory modalities varies across species, reflecting their evolutionary histories and ecological contexts, with diurnal visual predators relying heavily on motion detection while nocturnal species weight thermal and chemical cues more heavily. Prey discrimination, deciding whether detected items merit predatory investment, likely integrates information across modalities to assess prey identity, size, and vulnerability before committing to energetically expensive capture attempts. The neurobiology underlying these integrative sensory processes remains poorly understood in snakes compared to well-studied mammalian or avian model systems, though the fundamental challenge of constructing unified perceptual representations from disparate sensory streams remains similar across vertebrate taxa. The study of snake sensory integration promises insights into general principles of multisensory processing while revealing specific adaptations unique to snake neurobiology. The future trajectory of snake populations globally will depend on how effectively humanity addresses the interacting threats of habitat destruction, climate change, persecution, and international trade. The potential for positive outcomes exists if conservation efforts receive adequate funding and political support, protected area networks expand to encompass representative snake habitat, and public education reduces negative perceptions driving persecution. The alternative trajectory, where current trends continue unabated, portends widespread population declines and potential extinctions, particularly affecting habitat specialists and species with restricted distributions. The cascading ecological consequences of snake declines could include altered prey population dynamics, modified disease transmission, and disrupted food web structures, creating ecosystem impacts extending far beyond the direct loss of snake species themselves. The ethical dimensions of snake conservation invoke questions about humanity's responsibilities toward non-human life and the value we place on biodiversity independent of direct human utility, challenging societies to articulate coherent environmental ethics guiding conservation prioritization and resource allocation in an era of finite budgets and competing demands. Ultimately, the fate of snake populations will reflect broader societal choices about how we value nature and what kind of planet we wish to inhabit and bequeath to future generations.